Autism spectrum disorder, affecting 1 in 110 children, is characterized by language and social deficits as well as stereotyped behaviors. A microdeletion of 27 genes on chromosome 16p11.2 has been identified as one of the most penetrant risk factors for autism, present in up to 1% of autism spectrum disorder patients. Our lab created a mouse model of the 16p11.2 deletion to examine neuroanatomical abnormalities associated with this mutation using a Cree recombinase system. Preliminary data has shown abnormalities in dopamine signaling, which acts on higher-level behavioral control, motor learning, action selection, reward sensing, and motivation, many of which are impacted by autism spectrum disorders. In the striatum, dopamine modulates cortical input through two pathways, one direct and one indirect. Neurons expressing the D1 receptor act through the direct pathway and are associated with positive reinforcement learning. Neurons expressing the D2 receptor act through the indirect pathway and are associated with punishment and negative reinforcement. One of my projects was to quantify differences in D2 receptor expression between wild-type and mutant mice. Evidence from single-cell gene expression analysis and electrophysiology data indicated changes in medium spiny neurons, the primary cell type in the striatum. We hypothesized that changes in dopamine receptor expression and dendrite morphology could account for the deficits in dopamine signaling and increased corticostriatal synaptic strength seen in mutant mice. My other project was to quantify the number of dendritic spines on medium spiny neurons in mutant and wild-type mice. Using transgenic mice expressing GFP under the regulatory elements of the D2 receptor, we obtained brains from 3 16p11.2 mutant mice and 3 wild-type mice. After paraformaldehyde fixation, sagittal sectioning, and labeling of cell nuclei with Hoechst dye, we used fluorescence microscopy to image the striatum of each 20 micrometer thick slice. We chose representative sections from each dorsal and ventral striatum to quantify GFP expressing cells versus all cells present. We used the Golgi method to stain medium spiny neurons in the nucleus accumbens, then used Neurolucida imaging software to quantify the number of dendritic spines for 20 neurons, 10 from wild type and 10 from 16p11.2 mutant mice. Shown here are graphs of the total number of D2 GFP expressing cells in the dorsal and ventral striatum across 3 wild-type and 3 16p11.2 mutant mice. Changes in expression were significant in the dorsal striatum for all 3 pairs, with a p-value less than 0.01, but were not significant for the third pair in the ventral striatum. This graph represents the density of dendritic spines per micrometer dendrite in wild-type and 16p11.2 mutants. Results were not significant, with a p-value greater than 0.4. 2 out of the 3 16p11.2 mutant mice had significantly more D2 expression in both the dorsal and ventral striatum, supporting our hypothesis that dopamine signaling was disrupted. The range of disruption potentially parallels the range of penetrance in human patients, which varies from no defect to severe autism. Further research is required to see if D1 receptor expression is also changed, and if the change persists into adulthood. However, the number of dendritic spines was unchanged between mutant and control mice, suggesting that the increased strength of cortical striatal synapses is caused at the synaptic, not morphological, level. In order to study human phenotypes associated with the 16p11.2 deletion, we have been maintaining 6 induced pluripotent stem cell lines from human fibroblasts, 3 mutant and 3 controls. After designing a viral vector to fluorescently label each genotype, we plan to differentiate the cell lines into striatal and cortical neurons for gene expression and electrophysiological analysis to confirm the findings in mice.